to see the full screen? Students?
Yes, sir.
Yes, sir.
Okay, today we'll start with the stages of pneumonia. Okay, so today we'll start with the stages of pneumonia. Already we have discussed about classification of pneumonia. You know that pneumonia is nothing but this is an inflammation of a lung tissue, inflammation of alveoli. Okay, so how this pneumonia develops, right? How this all pathogenesis or the different mechanisms occurs, that we will discuss one by one, right? So for easy understanding how this pneumonia develop inside the body, the development of pneumonia is divided into total 4 stages. The first one is stage of congestion, second one is stage of red hepatization, third stage of gray hepatization, and the last one is stage of resolution. Okay, so first we'll discuss about stage of congestion. Stage of congestion, right? So name itself indicate here what happened. There will be congestion. Okay, so generally what happen when any infectious agents or when any etiological agents, when they enter inside the lung, when they enter inside the alveoli, ultimately they provoke inflammations, and all you know that whenever there is inflammation, ultimately as a first defense mechanism, there will be increased blood supply to that particular area, right? So due to this inflammation, ultimately what happens, majority of the blood vessels which are present near the site of infection, near the site of inflammation, that alveoli will show hyperemia, right? So ultimately what happens, there will be increased blood supply. Blood supply will be increased at the site of infection, right? So all we know that whenever there is an increase in the blood supply, ultimately what happens, when blood supply increased, ultimately hydrostatic pressure at that particular area will be increased, as well as due to inflammation, vascular permeability at the site will be also increased, so there will be exudation of a plasma, right? So plasma will be come out from the blood vessels, from these capillaries, as well as initial stage, small quantity of inflammatory cells like neutrophils are also come out from the blood vessels, right? So here you can see this is schematic diagram, right? This is alveolar septa, this is alveolar lumen. Right? So in response to any infection, in response to any inflammation, the blood supply to this particular capillaries will be increased. So ultimately there will be hyperemia, and due to excessive accumulation of a blood, the pressure will be increased, as well as due to infection, due to inflammation, or due to inflammation, the vascular permeability will be increased, and ultimately there will be exudation of fluid. This fluid which is present inside the capillary, it will come out, and ultimately it will go into the alveolar spaces, right? It will go into the alveolar spaces where these pathogens are present. In this case, in this particular picture, here you can see this pneumococcus. This particular pathogen is actually present inside the alveoli. Okay, so here what happened? Fluid as well as neutrophils are entered into the alveolar spaces, right? And generally, development of this required either 2 minutes or maximum up to few hours, right? So within 2 minutes to few hours, the stage of congestion will be developed, right? So grossly what happened, when blood circulation increase into the lungs, slowly and slowly the color will be changed, right? The normal color will be changed, as well as if you see the air spaces, normally this particular lumen is actually filled with the air, right? But slowly and slowly what happened, due to exudation of a fluid as well as inflammatory cells, ultimately the air spaces will be replaced by fluid as well as inflammatory cells. Now the second stage, after then what happened, the stage of red hepatization, right? Here there is 2 words, you have to remember this thing, red and hepatization, right? So normally what happened, in normal physiological conditions, lungs will be spongy, right? If you try to fill that particular lung, if you press the lung tissue, then they're spongy because ample amount of air is actually present in their alveolar spaces, right? And due to this reason, the consistency, the texture of the lung is spongy, right? And if you touch the liver in normal physiological condition, generally livers are actually firmer, right? They are little bit harder than lungs, right? They are not just like lungs. They will be, the consistency of the liver is not spongy, right? They are little bit firmer than lung. Okay, so here what happened in case, in a stage of red hepatization, what happened, we already see that the majority of the capillaries are get engorged with the blood vessels, their vascular permeability increased as well as pressure is also increased. So ultimately fluid as well as inflammatory cells are come out from the capillaries, as well as red blood cells. There will be extravasation, there will be diapedesis of, or sometime maybe due to hemorrhages. Ultimately this RBC which are normally present inside the capillaries will again enter into the alveolar lumen inside the air spaces, right? So here now you can imagine what happened. Most of the air spaces are actually replaced by fluid, inflammatory cells, and RBC, right? So no air spaces present, right? And if you touch such type of lung, then what happen? Normally, when air species is present, ultimately when you fill such type of lung, you will feel sponginess, right? But here what happen? Most of the air is actually replaced by fluid, inflammatory cells, and RBC. So ultimately, the sponginess will be lost, and ultimately the consistency becomes just like a liver, firmer than normal, right? Little bit harder than normal, right? So the name given hepatization, right? The name, why this hepatization name given? Because due to excessive accumulation of a fluid, RBC and WBC inside the lumen of alveoli, ultimately the consistency change and it becomes just like a liver. So the name given red hepatization, right? And why the red? Because there will be excessive accumulation of RBC. Okay, so grossly what you will find in case of red hepatization, grossly there will be consolidation of a lung, right? Okay, now what is consolidation? Consolidation is actually a typical term which describe the firmness or hardness of a lung, right? When the sponginess of the lung is lost, ultimately that particular part become little bit harder, little bit firmer than normal, and in that case we have to use the word consolidation, right? So etiology for the consolidation may be atelectasis. Whenever there is atelectasis, whenever there is a collapse of all the alveoli, in that case also what happen? Ultimately there will be a loss of air spaces and lungs become firmer or harder than normal, right? As well as whenever there is any exudate present inside the alveoli, either inflammatory exudate or exudate, right? In that case, whenever there is a fluid is present, whenever there is RBC is present, whenever there is a WBC present, in that case ultimately the sponginess of the lung will be lost and become harder, right? So in that case we have to use the word consolidation, right? So consolidation is a term which describes the grossly, it is generally used to describe the gross changes in which the lung loses the sponginess. Okay, and microscopically what you will find, macroscopically most of the alveolar spaces are actually filled with the neutrophils, macrophages, lymphocytes, RBC as well as fluid, right? Okay, and this time required almost 2 days for development. Okay, after entry of pathogen inside the lung, it will take almost 2 days for development of this red hepatization, right? Okay, so let me take a small, then this particular, in this stage lung piece will be float or it will be sink. What happen if you put such type of lung inside the fluid, inside the water?
Sink in what?
Sink in water, because there will be no air species present, because all the air species are actually replaced by the fluid, by the RBC or by the exudate. Okay, so in generally what happen, if you lift the lung, it become heavier than normal and it become more reddish. And if you take a small piece of the lung which is affected and if you put inside the water, in that case ultimately it will go to bottom because it will sink, no air species present. Okay, so here you can see this is gross picture. Okay, here you can see this is lung. Okay, and entire lung is become reddish in color. And if you feel the consistency in that case, because majority of the alveoli are replaced, air species are replaced by the fluid, by the RBC, by the WBC, so ultimately the sponginess will be lost and ultimately it become firmer, right? It become harder as compared to normal. As well as if you see, if you are able to see this marking, right, broad straps, right? So these straps are actually indicate they are actually represents the rib impression, right? So whenever lung expanded then normal, in that case ultimately this such rib impressions are present, and such type of lung, if when you open the thoracic cavity, in that case lungs will not collapse. In normal physiological conditions, what happen when air is not present inside the lung, generally when you open the thoracic cavity, slowly it will suck the air and ultimately entire healthy lung will be get collapsed, right? But here what happen, as there is no air species present, most of the air species are replaced with the fluid, with the RBC, with this WBC, hence such type of lungs will not collapse. Then next is stage of gray hepatization. Okay, so here what happened, this is little bit later in the development, right? So initially there will be congestion followed by red hepatization, and now slowly majority of the RBC which is present inside the alveolar lumens are phagocytized by the macrophages, and they are actually removed from the site, as well as majority of the other exudates will be removed by the macrophages, right? So ultimately in this particular stage resolution is actually started. Our macrophages are trying, our other inflammatory cells are trying to remove the exudate which is present inside the alveoli, and ultimately they are actually clearing the spaces for air, right? So grossly again here what happened, initially lung was reddish in color, but due to removal of the majority of the RBC from the site of infections, from the site of inflammation, ultimately the color will be again changed and it become slightly grayish in color or slightly paler in color, right? Because majority of the RBC are removed from the alveolar spaces, as well as slowly as time advance, majority of the neutrophils will be replaced by mononuclear cells like lymphocytes, plasma cells, as well as macrophages. And if are very or other fungal agents, in that case if macrophages are unable to kill that particular organism, in that case again macrophages will be become epithelioids, and lastly they are converted into multinucleated giant cells, right? So again in stage of gray hepatization, again still majority of the air species are not clear, so if you take a small piece of lung and if you put inside the water, again it will sink. Okay, and in last is a stage of resolution. However, here what happened, our macrophages do their job excellent, right? So ultimately majority of the exudate which is present inside the alveoli are actually completely removed, and ultimately again the spaces which are earlier occupied by the exudate, now that space can be utilized by air exchange, right? So there again air will be entered at the site. Okay, so here ultimately again lung will start to function normally, and this is known as stage of resolution, right? So this is typical pathogenesis of pneumonia. This is typical development of a pneumonia. Initially there will be congestion followed by red hepatization, gray hepatization, and finally resolution. Okay. Okay, so now we'll discuss about different type of pneumonias. So first we will discuss about bronchopneumonia, right? So name itself indicate bronchopneumonia. Here what happened? Both bronchi as well as alveoli are affected, right? So when both the components are affected, particularly bronchi, small bronchioles as well as alveoli, in that case we have to use the word bronchopneumonia. The hallmark of bronchopneumonia is actually the exudative lesions originate at the bronchiolar alveolar junction, right? Where this actually bronchi end and alveoli starts, at that particular point, initial infection will start. And slowly, and then this particular infection will enter inside the alveoli as well as it will go upwards toward the bronchi, right? So this both portions slowly they will affected. So ultimately, due to these both components are affected, we use the term bronchopneumonia, right? And particularly in majority of the cases of bronchopneumonia, generally the causative agents or etiological agents or pathogens are generally entered through air, right? So generally they are airborne route of entry of the causative agents for broncho. Another most, it is characterized by cranio-ventral consolidation, right? Cranio means anterior, ventral means downside, right? So anterior lungs are more commonly affected, and it become consolidated, right? So we have already know that what you mean by consolidation. Consolidation means when air spaces of the alveoli are replaced by either fluid, exudate, or sometimes it may be due to atelectasis, right? So when air space is not present inside the lung, ultimately lung consistency become firmer, harder than normal, and in that case we have to use the word consolidation, right? So here we have already discussed that initial injury will start from the mucosa of bronchioles and ultimately it will go down towards alveoli as well. It will go upward towards the bronchi. Okay. The major etiological agents in the case of bronchopneumonia are actually bacterial, right? So in our animals, most of the time bacteria are actually responsible for development of bronchopneumonia. Depending on the species, if you take example of a cattle and sheep, most commonly Mannheimia hemolytica, Stophilus somni, or Pasteurella multocida are responsible for bronchopneumonia in cattle and sheep. In case of swine, Pasteurella multocida, Actinobacillus pleuropneumoniae, Streptococcus suis, like bacteria are responsible for development of bronchopneumonia. In horses, Streptococcus, Rhodococcus, Actinobacillus, and Klebsiella are actually responsible for development of bronchopneumonia. In case of dog and cat, Bordetella bronchoseptica, Pasteurella, Staphylococcus, E. coli, and anaerobic bacteria are responsible for development of bronchopneumonia, right? So most importantly, bronchopneumonia in particular animals are mostly associated with bacteria. Okay, then predisposing factors. Predisposing factor means the factors which make animal more susceptible than normal, right? Here what happen? The animals become more susceptible due to certain things, right? Due to certain things, right? So suppose, for example, if you have anemia, if you have a low amount of hemoglobin, in that case you will get easily fatigue, right? So anemia is actually predisposing factor for your fatigueness, right? So here also what happen? Predisposing factors make more susceptible. Animals become more susceptible due to this reason, right? So here what happen? Some typical viral infections like bovine herpes virus 1, bovine respiratory syncytial virus, bovine parainfluenza virus 3, mycoplasma, some toxicity, right? So all these particular etiological agents like T2 mycotoxins, right? All these particularly toxins as well as pathogens, what they do, they actually impair mucociliary clearance, right? They actually impair the normal movement of a cilia. So ultimately, the pathogens are not clear from the site, as well as viruses like bovine herpes virus 1, as well as some other viruses of other animals. They actually interfere with the normal function of macrophages, alveolar macrophages, right? So when normal function of the alveolar macrophages actually not occur properly, in that case what happens? They will not able to remove the pathogens from the alveolar spaces, right? Particularly stress, toxicity, or viral infection, they will interfere with the normal ciliary movement, as well as they also interfere with the normal alveolar macrophage function. So ultimately it make animal more susceptible for infection, as well as this infection actually affect normal epithelium as well as alveolar septal tissue. It will damage that particular tissue, so damaged tissue are comparatively more susceptible, right? So due to all these particular reasons, these all factors actually predisposing the animals for development of bronchopneumonia, particularly bacterial bronchopneumonia. Okay, now bronchopneumonia are again actually subclassified into either suppurative bronchopneumonia or fibrinous bronchopneumonia. Suppurative bronchopneumonia, name itself indicate exudate will be, what happen? Exudate will be, in case of suppurative bronchopneumonia, which type of exudate you will find? Suppurative means?
First point.
Very good, right? So here what happen? In case of suppurative bronchopneumonia, we will find pus. While in case of fibrinous bronchopneumonia, the exudate will be fibrin, and this particular fibrin will be come out from the circulation, right, due to increased vascular permeability. Okay, so here in case of suppurative bronchopneumonia, exudate will be predominantly pus and mainly due to excessive infiltration of neutrophils. These neutrophils will release the lytic enzymes, and ultimately it will kill the bacteria, as well as they will also kill our tissue, and they will convert it into pus. Second important thing, the suppurative bronchopneumonia are generally more common in cattle and swine, right? And generally they will show lobular pneumonia. They will show lobular pneumonia. We all know that in case of cattle and swine, the lungs lobes are again divided into small lobules with the help of interlobular septa, right? So due to presence of interlobular septa, infections which occur in one lobules cannot easily pass into the nearby lobule, right? So generally few lobules are affected. So the pneumonia, generally in case of suppurative bronchopneumonia, we also use the term as a lobular pneumonia for this suppurative bronchopneumonia. Okay, in case of fibrinous bronchopneumonia, the exudate will be fibrin. And generally, in case of fibrinous bronchopneumonia, the etiological agents are comparatively more severe as compared to suppurative bronchopneumonia, and it will increase vascular permeability of the blood vessels, so this fibrin can easily come out, right? So the severity is more. So generally, rather than single lobules, entire lobe will be affected. Right. So due to this reason, this fibrinous bronchopneumonia are considered as a lobar pneumonia, as well as due to more severity of the pathogens, many a time pleura are also affected. Right. So pleura is also affected. So again, many a time this fibrinous bronchopneumonia is also called as pleuropneumonia, because due to as etiological agents are more pathogenic, it induced more severity of the lesion. So ultimately, entire lobe is lobar pneumonia, as well as due to more severity, due to more pathogenicity, ultimately they also affect the pleura, so the name given pleuropneumonia. So first we'll discuss about suppurative bronchopneumonia. Okay, so the suppurative bronchopneumonia is again same. This is bronchopneumonia, so there will be involvement of a cranioventral area of the lung. The cranioventral area of the lung become consolidated. And why these cranioventral lungs are affected more commonly in case of bronchopneumonia? So there are few theories why this cranioventral lungs are affected. First one is many a time what happens when this any pathogens, any particulate matters, when they enter inside the lung, there will be sudden changes in their direction. So ultimately, due to gravitational force as well as due to the multiple branching, that particular pathogens will be settled in this particular part, anterior part of the lung, right? And due to gravitational force, it will go downward side. So ultimately, ventral portion is also affected. So most commonly due to gravitational force as well as due to abrupt changes in their flow, cranioventral lobes are more commonly affected. And as we have already discussed, in case of bronchopneumonia, the pathogens are generally entered through inhalation route, through airborne routes, trachea, as well as probably there are some proper ventilation is also different from other areas. So this cranioventral part is more affected in case of bronchopneumonia. Okay, here what happens, the inflammatory processes are confined to the individual lobes, and grossly what you will find typical checkerboard appearance. What is checkerboard appearance? That we'll discuss little bit later on, so you can easily understand. Okay, and only small lobules are affected, so the name given lobular pneumonia. The etiology for suppurative bronchopneumonia are Pasteurella multocida, Bordetella bronchoseptica, Tropurella pyogenes, Streptococcus, E. coli, and many other microorganisms are responsible for development of suppurative bronchopneumonia. This suppurative bronchopneumonia is characterized by cranioventral consolidation of the lungs. And if you open the lungs, you will find definitely pus, as the name given, suppurative bronchopneumonia. So you will find the pus. And microscopically, majority of the alveolar species as well as bronchi are filled with the suppurative exudate, particularly neutrophil-rich exudate, as well as blood vessels will show hyperemia. In other cases, you will find infiltration of a lymphocyte, macrophages, and some other inflammatory cells. And name itself indicates this is bronchopneumonia, so bronchi as well as alveoli both will be affected. Okay, so this is actually a typical picture of a suppurative bronchopneumonia. This is in situ picture, and here you can see this is cross section of a lung. Okay, so here you can see this is this is anterior portion. From this actually trachea is enter inside the lungs, right? So this is anterior portion, this is cranial portion, and this is diaphragm, this is liver, so this is caudal portion. Okay, here you can see this particular area which is showing little bit dark red in color. Okay, they are depressed from normal surroundings, healthy lung. This particularly slightly yellowish lung is actually normal lung, marked by N, right? So this is comparatively healthy normal lung. And if you compare these two, you can easily appreciate that this particular lung is little bit depressed, as well as if you touch such type of lung, it will be more firmer, more harder than this particular normal, right? So this particular area is considered as a consolidation, right? And here you can see this is cranial part. Okay, so here more commonly this cranial portion is very severely affected, as well as here you can see this is ventral part. Okay, cranioventral. This one is dorsal part. Okay, when animal is in standing position, this will be go up, right? This is actually caudal part, and this is dorsocaudal area, and this is ventral area. Okay, so here generally this ventral area are affected, cranial part is more severely affected. So generally in case of bronchopneumonia, cranioventral portion is more severely affected. This particular organ is lung. Okay, and now you can see the cross section. Here you can see the cross section. The N means normal, right? Here you can see this particular lung, almost this white area is almost comparatively normal, right? But now you see this lower red portion is actually affected one. They are actually show some pathology. But in between these changes, if you able to see, here you can see this is one lobe which is almost like normal. Okay, then again there will be dark lobules. Okay, it become dark in color due to presence of RBC. Here you can see these are almost normal, while these are become reddish in color, right? So one lobule is normal, one is affected, one is normal, one is affected, one is normal, one is affected. So it will give typical checkerboard like appearance, just like chess. If you are playing the chess, in that case what happens, you will find multiple squares like white square and black square, right? So here also the same appearance is actually visible, white square and dark square. Okay, white square, dark square, right? So ultimately it will give typical checkerboard like appearance, right? So which term we will use for typical? Because some of the lobules are not affected, some of the lobules are affected, and they contain pus, so we will use the word suppurative bronchopneumonia. Okay, now we'll discuss about fibrinous bronchopneumonia. Okay, fibrinous bronchopneumonia, name itself indicate here what happened, exudate will be fibrin, right? And in this particular fibrinous bronchopneumonia, the etiological agents are actually more pathogenic. It will produce more severe effect, and ultimately it increased vascular permeability with more severity. So fibrin, which is normally present inside the blood vessels, they will come out, it will exude, it will come out from the blood vessels, and ultimately it will enter into the lungs, and it will be deposited into the alveoli and as well as surface of the pleura, right? So here, due to more severity, ultimately fibrin will be come out, and it will be deposited over the different lobe of the lung, as well as over the pleura, as well as inside the alveoli. And due to more severity, generally entire lobe is affected rather than individual lobules. Entire lobe is affected, so the name given lobar pneumonia, or as in this particular case, due to more severity, due to more pathogenicity, pleura is also affected, so the name given pleuropneumonia. Okay, the etiology for fibrinous bronchopneumonia are generally Mannheimia hemolytica, Histophilus somni, Actinobacillus pleuropneumoniae, Mycoplasma bovis, or some other disease like contagious bovine pneumonia caused by mycoplasma. Right? Agents are responsible for development of fibrinous bronchopneumonia. They are more pathogenic lesions, so it will affect entire lobe rather than lobules. Okay, grossly what happened? Grossly in early stage, as we know that there will be stage of red hepatization. So ultimately, due to severe inflammation, due to increased vascular permeability, fluid and RBC as well as WBC will be come out from the circulations, and it will enter into the lungs, into the alveolar spaces. So in initial stage, it become reddish in color, right? But slowly, as disease advanced, ultimately due to increased vascular permeability, fibrin will also come out from the blood over the alveoli, inside the alveoli, as well as over the surface of the lungs and pleura. And particularly in case of cattle and sheep, in case of cattle and pig, due to severe infection, due to severe inflammation, due to increased vascular permeability, the alveolar septa also get thickened. The interstitial septa, not alveolar septa, interstitial septa are get thickened due to accumulation of fluid as well as exudate, right? And this will give typical marbled appearance. What is marbled appearance? That will I will show you a little bit later on, right? As well as due to increased vascular permeability, fluid will be also accumulate inside the thoracic cavity, and this will leads to hydrothorax. And this particular hydrothorax fluids are actually contain enough amount of fibrin, because due to infection, due to inflammation, vascular permeability increase, as well as fibrin can easily come out from the blood vessels. Macroscopically, what you will find macroscopically, the fibrin will be deposited into the bronchioles as well as inside the alveoli, and this particular fibrin will be mixed with the edematous fluid, or it may be mixed with the inflammatory cells. Right, here you can see this is gross picture, okay, and here you can see this is comparatively normal lung. This is cranial portion, this is ventral portion, right? So this cranio-ventral portions are more severely affected as compared to caudal one. Okay, then here you can see, rather than individual lobules, entire lobes are affected, right? Entire lobes are affected. Here you can see this is entire lobes are affected, and these entire lobes are covered with this white capsule, right? Capsule-like material, right? So this is actually not a capsule, but this white material is nothing but they are actually fibrin, right? If you want to remove this particular fibrin, they may come out, okay, and underlying abnormal lungs can easily visible, okay. So this is deposition of a fibrin in particularly fresh cases. In little bit older cases, this fibrinous bronchopneumonia, this particularly fibrin, we have already discussed that fibrin will be replaced by fibrous tissue, and ultimately you will find scarring, right? Many a time this affected portion will get attached with the thoracic walls, or it may show attachment with some other tissue, right? So this is fibrinous bronchopneumonia. One entire lobe is affected, so the name given lobar pneumonia, as well as overlying pleura is also affected, so the name given pleuropneumonia. Okay, here is the marbling. Why we use the word marbles, right? If you see the white marble, right? શું હોય white marble ની અંદર તમને કઈ ટાઈપનો પેટર્ન્સ દેખાય? માર્બલ હોય આપણે જે નીચે કે નોર્મલ માર્બલ, એની અંદર તમને કઈ ટાઈપની પેટર્ન દેખાય એની અંદર? વ્હાઇટ માર્બલ હોય એની અંદર બ્લેક લાઇનિંગ દેખાય ને આપણને?
Yes, sir.
Okay, so just like that, right? Just like marble appearance. Marble appearance means if you see the white marbles, you will find typical line in between the marbles, right? So here same thing happen. Here you can see this is the normal alveolar, normal lung tissue. Sorry, this is lung tissue. Okay, and here you can see these are the lobules, and these particular lobules are separated by this thick yellow septa. This is actually interlobular septa. This is interlobular septa, and this interlobular septa is actually get thickened due to excessive accumulation of a fluid as well as inflammatory cells, and this particular affected lung tissue as well as this septa giving us typical marbled look, right? So the name given marbled appearance, right? So they are look like just like marble stone. Okay, here you can see this is another picture, little bit chronic picture. Here you can see this is one lobule, this is another lobules, this is third lobules, and these particular lobules are separated by thick, and this interlobular septa is thickened due to excessive accumulation of fluid, excessive accumulation of inflammatory cells, as well as edema. Okay, how this bronchopneumonia, how this resolution of this bronchopneumonia occurs? Okay, so generally in case of suppurative bronchopneumonia, the etiological agents are comparatively less pathogenic. So many a times suppurative bronchopneumonia actually heal almost completely clearly, right? But this Fibrinous bronchopneumonia, the pathological agents are more severe, they are more pathogenic, so you will not find complete resolution, complete healing, right? It will not resolve completely, right? And the particular in case of Fibrinous bronchopneumonia, you will find noticeable scar, right? Scar will be easily visible on the surface of a lung. Many a time, as we have already discussed, many a time you will also find adhesion, right? This particular tissue will leads to formation of a adhesion, because we have already discussed that initially what happened, there will be deposition of a fibrin over the surface. Our inflammatory cells like macrophages, they will try to remove that particular fibrin, but if within particular time, if these macrophages are unable to remove their fibrin from the that particular space, in that case, from the surrounding tissue, fibroblast will enter at the site, and ultimately they will try to replicate, and ultimately due to this replication, that fibrous tissue will be completely completely transformed into the collagen tissue with the help of fibroblast. Okay, then what will be the sequelae of bronchopneumonia, particularly Fibrinous bronchopneumonia? Right, as Fibrinous bronchopneumonia do not heal completely, it do not resolve completely, right? So ultimately what you will find, you will find alveolar fibrosis. Why this fibrosis occur, we have already discussed. Okay, then bronchiolitis obliterans, we have already discussed that bronchiolitis obliterans means they are actually polyp-like changes that occur inside the bronchioles. And sequestrum, sequestrum is actually special type of sequelae of a bronchopneumonia where necrotic lung parenchyma are actually enclosed in one capsule, right? Necrotic lung parenchyma are actually enclosed in one fibrous capsule. So ultimately, this is our body defense mechanism by which actually the pathogenic bacteria which is present inside necrotic tissue, they cannot travel to the healthy tissue, right? So this is one type of our body defense mechanism. And most commonly, this is seen actually infracted or necrotic lung tissue. Here you can see this is this is actually lung, which is initially previously it was affected by bronchopneumonia, and here you can see this is exudate or completely necrotic mass. This is enclosed in one fibrous capsule, right? So this is known as sequestrum. Here also, this is a high magnification. You can see this is one fibrous capsule, and this particular fibrous capsule is actually enclosing this particular mass. This is necrotic lung tissue, and this necrotic lung tissue may contain pathogenic bacteria. So to prevent exudation or to prevent transportation of this particular bacteria from this tissue to healthy tissue, ultimately these capsules are found by our body. Okay, so this is all about bronchopneumonia. Now we'll discuss about interstitial pneumonia. Okay, name itself indicate interstitial means here what happened in case of bronchopneumonia. Most importantly, which particular part of the lung is affected in case of bronchopneumonia? Which part of the lung is affected in case of bronchopneumonia?
Bronchi and alveoli.
Cranioventral.
Cranioventral, right? So both bronchi as well as cranioventral part are actually more severely affected in case of bronchopneumonia. In case of interstitial, here what happens? The injury or the inflammatory process take place at alveolar septa. At alveolar septa, and as we know, the alveolar septa or alveolar wall is made up of blood vessels, mesenchymal membrane, and they are lined by most commonly by type 1 pneumocytes or rarely type 2 pneumocyte. Okay, so generally this alveolar wall contain endothelial cells or particularly capillaries. Okay, endothelial cells, basement membrane, as well as alveolar epithelium, which is actually either type 1 pneumocyte or type 2 pneumocyte. And we have all learned that which pneumocyte, type 1 or type 2, have a regenerative capacity? Which pneumocyte
Type
can
2 pneumocytes.
Very good, right? So type 2 pneumocytes have capacity to regenerate. It can easily multiply, and ultimately it can convert into type 1 pneumocyte. Type 1 pneumocytes are actually differentiated cells. They don't have any capacity to regenerate, and ultimately whenever they die, that particularly cells will be sloughed, and ultimately type 2 pneumocytes will be replicates, and they will fill the empty spaces, and ultimately this type of pneumocyte will be converted into type 1 pneumocyte. Okay, so here what happened, the name itself indicates this is interstitial pneumonia. So alveolar septa or alveolar wall is affected, and the inflammation can be seen in any of these three locations: endothelial cells, basement membrane, or over the alveolar epithelium cells. Okay, and most importantly, in case of like bronchopneumonia, generally cranioventral part is affected, but in case of interstitial pneumonia, generally entire lung is affected. Entire lung is affected, right? So in case of interstitial pneumonia, entire lung is affected. Majority of the lobes are affected, but sometimes dorsocaudal aspect of lungs is more severely affected. But otherwise, majority of the cases, entire lung is affected. Another important thing, in generally corona, this interstitial type of pneumonia is more commonly seen, right? In this particular COVID-19 disease, in particular human, this pneumonia which is developed by this coronavirus is just like interstitial pneumonia. Okay, then in case of bronchopneumonia, the pathogens are entered into the lung through only air route, but in case of interstitial pneumonia, the pathogens can enter into the lung by either airborne route as well as through hematogenous route or through blood route. Okay, the etiology for interstitial pneumonia, the most common etiologic agents are virus, right? Like coronavirus, right? In case of human, which is recently going on. Okay, so generally the toxic fume or viruses are actually responsible for development of interstitial pneumonia. In case of animals, herpes virus, influenza virus, canine distemper, canine adenovirus, they all are responsible for development of interstitial pneumonia, right? Like canine distemper virus, they are pantropic virus. Actually, they have affinity for so many tissues. They can multiply in so many cells. So ultimately, it will produce interstitial pneumonia. In canine adenovirus, they have very high affinity for endothelial cells, so ultimately they will multiply in endothelial cells of the alveolar wall and it will produce interstitial pneumonia, as well as many other toxic materials, as well as some antigens which are responsible for provoking immune response. They are also responsible for development of interstitial pneumonia. Okay, grossly what you will find, first of all, when animal is actually suffered from interstitial pneumonia, here generally what happen? When you open the thoracic cavity, the lungs will not collapse at all, right? Lungs will not collapse. When you open the thoracic cavity in normal physiological condition, a healthy lung will get collapsed. It become very small in size, almost half in size. Then when they are present inside the thoracic cavity, when the thoracic cavity is not open, when the thoracic cavity have negative pressure. Okay, but whenever the lungs are affected, in that case it will not collapse at all, because the sponginess of the septal wall is actually lost. Sponginess of the septal wall is lost ultimately, right? Due to this reason, it will not get collapsed. As well as on the surface of the lung, you will find typical rib impression, what we have already seen in few earlier images. Okay, then another important thing, in case of bronchopneumonia, generally the bronchi and alveoli are filled with the exudate. In case of suppurative bronchopneumonia, you will find pus as exudate. In case of fibrinous bronchopneumonia, generally fibrin is present. But in case of interstitial pneumonia, visible plus visible exudate, grossly visible exudate are actually not found, right? So this is another important difference between the interstitial pneumonia and bronchopneumonia. Okay, then depending upon the stage of development, in early stage lungs become reddish in color, but in later stage, when majority of the RBC are removed, in that case lungs become paler in color. As well as due to loss of sponginess, ultimately it become more elastic, more rubbery, and more meaty. Due to excessive accumulation of a fluid inside the alveolar space, ultimately lung become heavier also than normal. Right, so here you can see this is a typical picture. This is cranial part, this is caudal part, this is dorsal, this is ventral, and here you can see this almost entire lung is uniformly affected. As well as if you are able to see, some rib impressions are also present over the surface of the lung. Okay, rib impression is present over the surface of the lung, and here you can see this is cross section of the lungs. And if you touch, if you press such type of lung, it becomes slightly meaty, just like the muscles like meaty. Okay, and the sponginess is also lost, and they are also heavier than normal. Okay, here you can see the entire lung is affected. This is cranial portion, this is caudal portion, this is dorsal one, this is ventral one, this is heart. Okay, and the lung color is reddish, as well as if you see the rib impressions are present, it means they are showing that the lung was markedly enlarged when animal was live, right? As well as this is actually non-collapsed lung, the lung is not collapsed properly, as well as color is reddish, right? So when we found reddish in color, what it indicate? The time, this is actually acute changes or recent changes or little bit chronic changes,
Acute changes.
right? Acute changes, right? Whenever see reddish in color, it means the hemorrhages are comparatively more recent. But in particularly older cases, what happen when this RBC are completely removed by our macrophages and other mononuclear cells, ultimately, particularly inflammatory cells, in that case, lung become paler in color. Here also you can see typical, right? Lung is non-collapsed. Okay, here you can see this is anterior portion, this is posterior, this is dorsal, this is ventral, and lung color is completely off-white, right? So severe durations, acute or chronic,
Chronic.
right? Okay, because majority of the RBC are actually removed from the alveolar spaces. Okay, good. Okay, then interstitial pneumonia is again subclassified into two subtypes. First one is acute interstitial pneumonia and chronic interstitial pneumonia, right? So first we'll discuss about acute interstitial pneumonia. Name itself indicate acute interstitial means this is sudden change. Okay, this is sudden change. And in acute interstitial pneumonia, again they are divided into two phases. Acute interstitial pneumonia are divided into two phases, just like corona. Okay, in COVID-19 the same mechanism occurs. So this acute interstitial pneumonia are again subdivided into exudative phase and proliferative phase. Name itself indicate exudation, right? So here what happened? Due to increased vascular permeability or due to damage of the vasculatures, ultimately there will be exudation. And here what happened? Generally fluid as well as plasma protein will be come out from the circulation into the alveolar spaces. These particularly plasma proteins are mixed with some materials like lipids and other surfactants and other pus-like materials, and ultimately hyaline membrane, which is get deposited over the surface of alveolar wall. Okay, so ultimately what happened? The alveolar wall become thicker, it become thicker, as well as air exchange will not occur properly. Air exchange will not occur properly, right? So in acute interstitial pneumonia, first what happened? There will be initially there will be exudation. So due to increased inflammation, there will be increased vascular permeability. So there will be exudation of a fluid as well as plasma protein. These plasma proteins as well as fluid they will get mixed with the surfactant which is normally present into the alveoli, as well as some lipid materials, and they will form typical hyaline membrane. Hyaline membrane is nothing but this is actually macroscopic word to describe eosinophilic amorphous proteinous materials, and these particular proteinous materials are attached or they get deposited over the alveolar septa, and ultimately they will prevent normal air exchange. As well as due to this reason, the normal sponginess will be also not seen. After exudative phase, there will be proliferative phase, and in proliferative phase, what happen? After a few days, as due to pathogens, due to etiological agents, the type 1 pneumocytes are get damaged, they are get died, and type 2 pneumocyte will be replicate, and ultimately you will find typical proliferative phase. So in proliferative phase, you will find typical proliferation of type 2 pneumocytes, which actually replace, and ultimately this particularly type 2 pneumocyte will be differentiated into type 1 pneumocytes, and they will try to uh, heal the damaged area. And alveolar walls are thickened as compared to exudative phase because of more and more amount of hyaline membranes are get deposited. Right. So here you can see this is schematic diagram of acute interstitial pneumonia. Okay. So here you can see this is wall. Okay. So this is wall of alveoli. This one is capillary. This is type 2 pneumocyte, and this squamous epithelial cells are type 1 pneumocyte. Okay. So initially what happened? Whenever there is infection, whenever there is inflammation, ultimately the vascular permeability will increase and there will be exudation of this fluid as well as inflammatory cells into the alveolar septa. Here, this is alveolar septa, right? So due to exudation, ultimately this alveolar septa become thicker in size. As well as when this type 2 pneumocyte will damage, ultimately this type 1 pneumocyte when get damaged, ultimately type 2 pneumocyte will be replicated and you will find typical replications in proliferative phase. Then the second type of pneumonia is chronic interstitial pneumonia. So in chronic interstitial pneumonia, what happens when pathogens persist for a very longer period of time? In that case, the acute condition is converted into chronic conditions, and here slowly the fluid and the fibrin which is present into the alveolar septal walls are actually penetrated by the fibroblast, and ultimately there will be fibrosis is actually occur. Right. So generally what happens? Alveolar septa, which is normally showing sponginess, elasticity, ultimately they are replaced by the collagen tissue, ultimately replaced by the fibrous tissue. So they lost their elasticity and it become more firmer than normal. As well as there will be hyperplasia of type 2 pneumocytes, and sometimes you will find squamous metaplasia also. Right. So here you can see this is schematic diagram where you can see this is a capillary lumen, and if you compare earlier one, you can easily see that due to fibrosis here this particular septa is showing severe fibrosis, right? So due to excessive deposition of collagens, ultimately the lumen of capillary is also decreased, and the alveolar septa is actually infiltrated with the plasma cells. Here you can see this is plasma cells. Why these are plasma cells? Because they have ascending nucleus. Okay, as well as they are also penetrated by the fibroblasts. These fibroblasts will be multiplied, it will deposit the collagens into the septa, and ultimately the septa become thicker, right? So you will also find some neutrophils as well as eosinophils at the side, some macrophages. Okay. Okay, so this is about interstitial pneumonia. Today we'll stop here. Okay.